Hello. Today I will discuss the development of recombinant Pichinde virus vectored vaccine against Turkey arthritis reovirus. Turkey arthritis reovirus causes clinical lameness due to tenosynovitis. 35% to 70% of the birds in a flock are affected, causing huge economic losses, mainly due to culling at the marketing age. No commercial vaccine is available except autogenous vaccines. The cases of turkey lameness caused by turkey arthritis reovirus continued to increase even with the use of autogenous vaccines because these reoviruses are evolving fast, creating new divergent strains. That's why we need to update these autogenous vaccines regularly. Till date, no live vaccine is available for priming breeder turkeys before boosting, so we need a newer vaccine that can combat variant reoviruses. We came up with an idea of using recombinant Pichinde virus based vaccine vector that have three viral genomic RNA segments. Two of its genome segments have multiple cloning site where two foreign genes can be inserted. We used S1 and S3 genes of Turkey arthritis reovirus because they are responsible for formation of cell attachment and outer capsid proteins. We have formulated different combinations of monovalent and bivalent vaccines. Three strains of Turkey arthritis reovirus were used, and S1 and S3 genes of these strains were PCR amplified and cloned into the vec and the plasmids, and S1 and S3 genes of one of the isolate were codon optimized and then cloned into the plasmids. The clone was confirmed by RE digestion as shown in this figure, where the gel purified products were Sanger sequenced for checking mutations, correct reading frame, and orientation of our insert. These plasmids then were used to transfect BSRT75 cells in different combinations of monovalent and bivalent vaccine. In monovalent vaccine, GFP expression was observed at 48 to 72 hours post-transfection, suggesting successful transfection. Supernatants from BSRT7 cells were collected to infect BHK21 cells, where strong GFP expression was observed after 24 to 48 hours of infection, indicating rescue of viable virus, as shown in the figure B, which is then further confirmed by RT-PCR using S1 and S3 gene-specific primers. These vaccines were then further tested by direct fluorescent antibody test for the expression of reovirus antigenic proteins. Four different vaccines were used to infect BHK21 cells with one wild virus as positive control. The result shows that the bivalent codon optimized vaccine showed highest fluorescence, suggesting highest expression of reoviral antigenic segments and formation of antigenic proteins. These four viruses were then further inoculated into four different groups of turkey poults, where first dose was given at 1 week of age and boosted at 3 weeks of age. Before booster dose at 3 weeks of age, the monovalent S3 vaccine and the bivalent codon optimized vaccine showed higher antibody titer. And after booster dose at 5 weeks, the bivalent codon optimized vaccine showed higher serum neutralization antibody titers. The vaccine and vector was found to be safe and efficacious because none of the vaccinated birds shows any sign of clinical illness or any gross lesions at necropsy. In conclusion, I can say that bivalent codon optimized vaccine was most efficacious in vitro and in vivo, and in future work, we plan to test the efficacy of this codon optimized vaccine in vivo against heterologous and homologous virus challenge in turkeys. At last, I would like to thank RARF for funding this study and Indian Council of Agriculture Research for supporting my PhD. Thank you.